Greetings of the day, everyone. Hope you have joined the Telegram forum and also Classroom. If you haven't joined it, please do join immediately. The link is available in the mail and also in the guide. Dr. Gasala, shall we begin? Gasala, you're not audible. Can you please be a little bit louder?
Yeah, shall we start the session?
Yes, please begin.
Okay. Okay, so good evening, everyone. Let me share the screen so that we can start the session accordingly. So today we have the session for prerequisite number 2, where we will be discussing about gene expression and types of data that we are going to deal with and the databases from where we will be retrieving the data for analysis. Please slide. Okay, so I'm not sure. So could you please give me in chat that how many of you are already aware of gene expression data? If you have previously worked or it is totally new for you? Okay, so okay, what about others who are in the meeting right now? Okay, so we have more number of beginners than the experienced ones. Okay, so we shall start with very basic. Let's get into it so that we can understand how exactly the data is and from where the data is exactly actually coming from. So we all know the central dogma of biology that within each cell there is nucleus which is composed of chromosome pairs, and the chromosome is composed of or the nothing but the having the sequence of DNA, and on that DNA there are segments that are dedicated to a specific gene. So there are multiple genes on the DNA segments. These genes and their corresponding gene expressions we are going to analyze, because whenever there is a disease happening in your body, there will be certain alteration or changes at the DNA level. So from DNA, after transcription and translation, we obtain mRNA and then we obtain proteins. So at any, like at the lower or the more deeper level of biology, we get these changes and we try to understand that how these changes are impacting your body to go towards the diseased stage rather than in the healthy stage. Okay, so for analysis or for dealing with this type of data where we are trying to understand the gene expression data, we need to extract the samples from samples, and samples are composed of DNA sequences. So what we do is it is extracted from the patient who is being diagnosed with certain disease or having probability of having certain sub disease. Once it is extracted and in wet lab experiments, library is prepared for each segment of your DNA will be mapped to the library, whatever DNA library which is already predefined in the wet lab setup. And then further steps are added also within wet lab, that adding of adapter sequences to stabilize your extracted DNA sequences. Okay, because when DNA is inside your body, it is totally in the stable form, but when it is extracted outside and in lab right now, okay, so in petri dishes, so in that case it may goes into unstable stage. So for to stabilize those sequences, so that we can capture their values, expression values clearly or accurately. We try to stabilize them by adding adapter sequences and poly A sequences for two with our DNA sequences that we have fragmented. And after that step, we have the fourth step, which is the sequencing step. So here we have different type of sequencer based on different generations. Like we are, if we talk about next generation sequencing, then we have sequencers like Illumina and others, which provide high throughput sequencing results for the sequences that we provide them after library preparation. Okay, so third step is sequencing, where we provide the sequence to sequencers, and sequencer will assign the sequence. First it will read the sequence which is there in your on your DNA fragments, and then it will assign the quality score to your sequences, so that we can understand that what is like how good the sequence is captured from the patient sample. Once we have the sequencing results, then we will be having the files in different format. Okay, so we actually have BCL format that is directly coming from sequencers, that is converted into FASTQ format, the BCL format, and then stored in the different databases respective to each disease. If there is disease specific database, otherwise they can be there in any open repository, that is primary repositories or secondary repositories like NCBI and Array Express and other repositories. So once we have the FASTQ files, we can further proceed for analysis of this type of data. So raw data will always, whenever I will be referring ahead in the course, for the raw data, I will be talking about FASTQ files. Or if we do microarray analysis, then we will be referring to cell files, because when we perform microarray analysis, so their raw files are cell file format, not the FASTQ file. FASTQ files are specific to RNA sequences or DNA sequences that are coming from Illumina. Okay, so there are further analysis, and this steps of analysis for converting FASTQ files into BAM file format and then converting BAM file into VCF file format. This is totally depending on the objective of our work and which pipeline that we want to follow for further analysis of our data once we have it. Okay, so hope that is clear that what exactly from where exactly the data is deriving. Now we will discuss the file formats. Here I have mentioned the first and most basic file format that we discuss in genomics data analysis is FASTA file. FASTA file will be having the extension of either .fa, you can see here .fa. So file name will be there, and then .fa extension could be there, and also there could be extension of .fasta. Okay, so both can be there and both will be having the same file format. So this is the basic description that FASTA was developed from, declared by William R. Pearson, and it is the first file format that was mentioned, where the sequences will be arranged in particular order that they have been retrieved from the samples from the patient or even from the healthy people, if we are trying to compare the sequences. So this is basically the figure, which is the second figure here. So here we will be looking at that FASTA file have two lines of code. The single FASTA sequence is having two lines, and always first line will be the header that will start with greater than symbol. Okay, so header file will be header line will be having the information of your sequence, that whether this sequence is from which organism or which sequencer it is captured, this particular sequence is captured, and from which which library they have used for this particular sequencing. Okay, so library we will understand further when we are going deeper into understanding this type of data. When once we retrieve the data from databases, I will explain you. Otherwise, you need to understand that FASTA file format will have two major lines. First line will be the header line and second will be the sequence line. Okay, so sequence line will be having the ATGC sequences that are there on your DNA. So, and if the FASTA file have more than two headers and corresponding sequences to them, then it can also be called as multi-FASTA file. Okay, and that will also having the extension of FASTA.fa or file name with .fasta. Okay, so we can have either just one header and one sequence in the FASTA file, or we can have more than one header and corresponding sequences in the FASTA file. But two lines should be mandatory. If in your FASTA there is no header line present, just the sequences, we need to have either we need to retrieve the right file format from the database from where we are downloading the data. Otherwise, if it is not, then we need to manually add the header line. Otherwise, the tools that we are going to use for which are bioinformatics analysis tools specifically, so they will not be able to read your data if header lines are not there, because this file will not be recognized as FASTA file. So we need to have the header line. That's about FASTA. And here is multi-FASTA. So that is the same multi-FASTA that I just explained. And multi-FASTA can have the either it will have .fasta extension, like same extension as single sequence FASTA file, or it can have a specific type of extension which is MFA. M stands for multi and FA is, we already know, for FASTA. Okay, so either the file can have the FASTA type of extensions for the file name, otherwise they can have a specific extension which is MFA. And if the file name is having the extension FASTA or FA or whatever we have all included here, if it is there, then we can understand that there are sequences within this file in FASTA format. Okay, if we have many sequences and the file is extension is still FASTA, then that is fine. Okay, we can call it FASTA file. But if the FASTA file is having more than one sequences and it is having extension of .mfa, it means it is always be having more than one sequence. Okay, and more than two headers corresponding to each sequence. So .mfa file, the multi-FASTA file, cannot have only one sequence. That will give error if you provide this file with just one sequence as a MFA extension. Okay, so hope that is clear that we have the FASTA file in the first category. That is need to be understood, that what is the order or the format of this file, and the extended file format is multi-FASTA. Next is FASTQ file. So FASTQ file is the advanced version of FASTA file where we have more information about the sequence. FASTQ file will be having 4 line of codes where the first line will be the header or the label, we can say label for the sequence. So whatever in the, and it will start with @. Okay, so this header line will be having same information that we discussed in FASTA file. For the instrument detail or the sequencer detail, then there can be detail of the organism, that from which organism this particular sequence is deriving, as for example we can say Homo sapien. And then the next line will always be having the sequences, the ATGC sequence that we already have in FASTA file. But the third line will be the plus sign symbol. Always this will be alone on the third line. Okay, so there will be nothing with plus symbol. And course after that, the fourth line will be the Q score of each nucleotide that we are having in the sequence. These Q scores are basically the ASCII scores that we have calculated with the formula that we have Q as the quality value, and we can also say as quality score, and with the formula -10 log 10 and P, probability of error. So what is the probability of error if the sequence that is captured is captured like wrong or misinterpreted by the sequencer. Okay, so accordingly, for each nucleotide that is captured by the sequencer, the scores will be assigned, and these scores will be deciding that whether your genes are more expressed or less expressed, because whatever captured by the sequencer, whatever sequencer read from your sequence, or the quality is read by the sequencer, that will be deciding your final gene expression. But right now in this form, as a ASCII characters, we cannot understand that what is the gene expression, right? Because these are not in number form or in general or the common format. Okay, so we need certain tools to convert these expression and corresponding to gene specific segments. Okay, so these whole sequence that is shown here in the figure is not corresponding to just one gene. Okay, maybe part of it corresponds to one gene and other part next to it is corresponds to other gene. Okay, so gene specific quality scores will be deciding the gene expression for that particular sample, which is here. So one complete information in FASTQ file will be for one particular sample in your data. And FASTQ file can have more than one sequences, and the next sequence will also following the same format. First will be the header file or the labels, second will be the sequence, and third will be the plus symbol, and then we have the quality scores corresponding to each nucleotides. Hope this is clear. And below this is an example where we are showing that what specifically each component in this header file mean here right now, because there can be various information corresponding to what sequencer you are using or what important information you want to keep in your header line, so that in future if you are going through your data, you should understand that this particular sequence is deriving from which particular location or where or on which sequencer it has been sequenced. Okay, so there is an instrument name that will be the sequencer name. Then we have the flow cell info. That is again the setup that is being done for your instrument, because on the same sequencer or the same instrument on which the sequencing is being done, we can modify the setup so that accordingly the sequencer can capture the sequences and their quality scores based on this. Okay, so further other informations are also related to the instrument that we are using and what setup we have used there based on the clusters, like cluster of like number of nucleotide sequences from here. For example, if we fix that, take the cluster of 15 nucleotide sequence at once and then assign the quality score, so that if the quality score are assigned in small chunks, maybe that will be captured more appropriately. Okay, so and the next line is the raw sequence letters that we already know from where it is coming from, directly from the DNA, which is extracted from the patient. And we have the quality values on the fourth line. So never forget the third line, because it seems very simple and unnecessary, but it is very necessary to complete the format of the file. Otherwise, your tools will give error and not provide any result, even if you have the right file and right information. Next file is BCF file format, which is known as variant calling file format. This file format, which usually we use when we perform mutation data analysis, where we try to see what mutation happened in the patient's gene level or at the protein level, maybe, that is causing the disease. Okay, so here below you can see here that if we see that actual sequence is ACG, but it is altered to either, sometimes it is captured by the sequencer that it is altered to A, the whole sequence is replaced by adenine. Otherwise, sometimes it is also captured that it is three nucleotides are replaced by just two nucleotides, which is adenine and thymine. Okay, so AT sequence. This is called mutation, where the changes are happening at single nucleotide level. Similarly, you can see another example where the adenine is a reference nucleotide, but it is altered to G, that is guanine, right? So this is called mutation, which is where the reference or the actual nucleotide is changing or altered to another nucleotide, which should not be there in normal human samples who are functioning normally without any disease. Okay, so if this changing is happening, this is not mandatorily mean that this is causing the disease. Okay, but they may cause the disease. So we need to further go into downstream analysis to understand the biological pathogenesis or biological functioning of this particular changes. If it is happening in the body, what will happen, what will be the consequences of these changes, and then only we can declare that this particular change in the body is causing the disease that we are studying right now. Okay, so this is about variant calling file, where on the top there will be header lines. So here there is not only one line for header but multiple lines. Okay, and every line is important because that is again declaring the details which is important for the file or the tools to understand that on which VCF tool version that it is like analyzed, and then which tool we have used. For example, we can say here we have, it is mentioned that VCF tools is used for to get this result from your sequencing data that we have in hand. And further other informations is also given related to the sequence, related to the format. We can see there are different informations based on all the types of details. So most of the time you will not be like explaining or understanding the header, because header will always be there in your VCF file, and it is important. Without header, your VCF file will not work. So even if header is not there, you need to provide with double hashtags the header by yourself. At least one line of header is important. But if the more information is there in VCF file, more better for your data. But if not there, then to provide in any VCF analysis tool, you need to provide at least one line of header that is for file format. You can mention that which file form VCF version, file format version is this one. Okay, and then we have the body, which will be in tabular format. So where in body it will be mentioned with different columns. First will be the on which chromosome this particular alteration is happening, and then the position on the chromosome, IDs of this particular mutation or the changes which is happening. For some IDs it is not mentioned because it is not identified by the tool on which you have done this mutation analysis. Okay, so whatever is already there on the tool reported and documented, then on for them only the IDs will be mentioned. Otherwise you need to explore through the literature survey. Okay, and then there will be the quality score and filter pass, that whether this particular filter is passed or not passed, and that will be the based on the tool that you are analyzing, and whether this particular type of mutation or changes is already reported on that or not. Okay, similarly there will be different other columns that will be there within your VCF file, and the here at the end you can see the sample information is there. So in this example is from where only two samples were considered, and similarly the data points and the number values are given based on the reference alleles and the alternate alleles, that whether the reference allele is GT, then it will be mentioned as 0. If the alternate allele is more than 0, then it will be mentioned as 2. So this is the technique that will be handled by the mutation analysis tools. Okay, so you don't need to worry or understand about these particular numbers. You just need to focus on your chromosomal location, the first 4 or 5 columns that we are having here, where we need to understand chromosome location, position IDs, reference nucleotides, and the alternate or altered nucleotides, so that you can see that what type of mutation has happened and whether these mutations are involved in the disease or not. Okay, so this data is also coming from your sequencing data, and we may use this in any type of analysis based on our objective. Next is the SAM tool, which is sequence alignment tool. Once we have the sequence in our hand, then we can convert, like for example, we have the FASTQ file, right? So FASTQ file will be having the sequence and corresponding quality scores of the sequences. That FASTQ file can be further aligned to the reference genome. Reference genome we call the sequence of the human genome, which are in standard format, where all the genomes are composed from healthy people. Okay, so no disease. Okay, so we will try to map or align our sequence, the FASTQ file sequences, to the reference genome, and based on that the SAM file will be generated. This SAM file will be having the header as a metadata that will be having the information of your sequence and other related information, whatever is relevant for your sequence, so that it can help to understand you the sequence. And next, it will follow the tabular format, where you will be having the records, as we have mentioned here, that there will be the read name. Read name is basically the sequence or the read that the sequencer has read from your sequence. So this will be the chunks of code or the DNA sequences on your in your sample. Okay. Next will be the flags that will be mentioning whether this particular read is passed or not based on the sequencer that we are using. Then we have the position information on the chromosome, and furthermore we have more information, so mapping quality and other informations. We have read sequences, we have the sequence for which the ID is given in the first column, for that the read sequences is given, and quality scores are given. So this quality score you are already having in your FASTQ file, but now you have more information, because right now you have the position information as well. Earlier you just knew what is the sequence from the FASTQ file, what is the sequence and the quality score, but now you also know what is the position of that sequence on your chromosome, and also the metadata of your sequence, that from which organism it is deriving. That was also there in our FASTQ file in the header form, right? So that can be added here in the metadata column. So after FASTQ file, we are having the SAM file that is more having more information or more comprehensive details of our sequence. From the SAM file, we can convert the SAM file into the BAM file, because SAM file is the tabular format, but BAM file is a binary alignment format. Okay, so binary format file which with the extension .bam, okay, and not in capitals. I have just mentioned here so that it can be highlighted here, but it will be in lowercase bam. This file will be helpful for us because it will help us to visually see that what is happening in our sequences, where the sequence or the nucleotide is captured like higher or more, and where the frequency of the nucleotide that is captured is lower, based on which the quality scores are assigned for our data. And again, it will be having the similar type of information, but now in the uh, binary format. Okay, so here below is the SAM file format, but we are converting this SAM file format into the BAM format to visually see what is already mentioned in the BAM file. Okay, so BAM file, we are seeing the position of our sequences, and sequences, read sequences will be always corresponding to certain genes. according to their positions. Okay. So we will be visualizing that information through figures, and we have different tools such as IGV and other tools. Genome browser is there to visualize. Okay. Next is the bed file format, which is browser extensible data file format. This file will be having the extension .bed. Okay, always .bed, and it will be having the tabular information with the data of chromosome start coordinate of that particular gene, that from on the chromosome 1 we understand, but what is where exactly on chromosome 1, that is the start coordinate, and end coordinate is where exactly on chromosome 1, or next to chromosome 1 is chromosome 2, so where exactly it is ending, so that exact position will be mentioned. So more accurate information, right? Or more precise information. Similarly, other values will also be added in bed file format. And this file we can also use to visualize the particular information that we have in our data. Okay, so this is the visualization of our data, where we can see that we have the samples on the left side, like see here. On the left side, we have the samples uploaded, and on below, like there are two tables, two boxes here in below section. We can see the first one is our sample, and second one we can call as a reference sample maybe. So if we compare, then in the reference sample, like in this particular position, we see that there is no particular read, there is no particular information here. If we consider it as a healthy sample, then there is no particular information. This particular gene has no corresponding sequences, right? But in the diseased sample, if we compare, then there are certain sequences available or certain information available in this particular gene. So there is variation, right? When we compare from disease to healthy samples, there is variation. And now we can understand, we can say that if there is a disease, then when the section or the gene where there should be no reads available or no sequences show any like information in healthy person, then why in the diseased person there is information, there are sequences or the reads captured by the sequencer, and this could be the reason for causing the disease. So this is not only reason, but this could be reason. Okay, so that like in the previous slide, as we saw that ATGC sequence is altering into just adenine, just A, or maybe just AT. Okay, so all the three nucleotides are changing by just one nucleotide. Okay, so here we see that if we have the adenine available here in diseased person, but in healthy it should not be there, right? So presence of adenine at this particular on this particular gene at this particular location could be the reason of the disease. Okay, so this is better way to view your data and to visually see that where exactly the problem is happening, what could be the possible reason for the changes that you are studying or for the disease to happen in the body of the samples that you are having. Next is gene transfer format, which is also called GTF format, and the extension will be .gtf. So if you remember the file names only, you will know the gene extension of each files, because extensions are important to be recognized by the tools which are available to handle this type of data. And GTF file format will again having the information of sequence ID, where the chromosome numbers will be assigned, and then there will be the information of type of that particular section on which that particular gene is residing. So within that gene, whether that particular sequence belongs to exon regions or it is from coding sequences. Okay, so more specific information than the previous files, right? So all these type of files will only help us to identify more insightful results from our data. Apart from GTF, we have GFF file format, which is general feature format, or we can also say gene finding format. Here we have the table in same format. There is just one changes in this file, which is score phase. So here we have in GFF file, we have the strand information, which is an additional column in GFF file format. Okay, so strand information is also important if we are studying about the gene expression and the position of genes. Okay, so sometime when the gene is from 5 prime to 3 prime, then it will be called on having on positive strand. If the sequence is from 3 prime to 5 prime, then it will be called as on negative strand. Okay, so strand information is there in GFF file format, and extension, that we have .gff2 or gff3 or just GFF is also there. It's hiding behind the table. So these file formats are there which can help us to explore data in more detail. CDS, okay, so if since most of you are new here, so CDS is coding sequences of your on your genome or on your sequence, DNA sequence. Coding sequences are those sequences which will be further translated into protein that will be functional part in your body and that will be playing different functions in your body, because some not all parts or all the regions on your gene will be the coding sequence regions. Okay, there will be non-coding regions also which will not be translated into protein. Okay, because we know that proteins are the most basic or like modules that actually work in the basic functioning of the body. So CDS, most of the time we focus on CDS if we are studying the coding sequences or proteins related information. Otherwise, we can also focus on like exonic regions and other regions. So we will come along with this type of data as well if we are dealing with RNA data. Next type of file format is cell file format. Okay. So, okay, before this, let me mention that, okay, let me know that if you all are following these file sequences, because theoretically it must not be very much clear, but at least I am adding the format of the files so that you can understand how exactly they looks whenever you are going to open your file, because most of the time we don't open the file. But if there is error or if you want to cross check the file format, then you should open your file and look at the format. Maybe there is some mistakes in the format. There is a header missing in your file. That's why your tool is giving error, right? So we always need to check the file format if there is any sort of error in the like in the output or in the results from the table. Okay. So are you all following the lecture? Is it clear, the file formats that we have discussed so far? Let me know if it is fast, then I can also like explain in more detail, so that where are you all? Like I cannot see anything in the chat box. Can you please give real quick yes so that I can
Dr.
understand?
Basava, we receiving thumbs up here in Zoom.
Sorry.
We receiving thumbs up in Zoom.
Okay, that's
Thanks.
clear. So good.
Yeah.
That's really good. And we will proceed with cell file format. So cell file format basically stands for CEL that we have in the cell intensity file. And these files will be having the probe intensities in our file. So it is it corresponds to specifically for microarray type of data where the data is generated from Affymetrix chips. Okay, so there are different platform as well in microarray itself, like Affymetrix. Then there is Agilent platform. Okay, so based on those platforms, we get different file format. Even in cell, in microarray, we have different file formats. Apart from cell, we have GPR file format. We will see when we are doing analysis or retrieving data, that how to check that which file we need to use and when to use. Okay, so these cell files will be having the information of the genes which are already been declared or reported in the different studies, okay, or in laboratories. So these are also extracted from either the diseased or the healthy samples. And then once they are extracted, so from DNA, RNA will be extracted and labeled. Based on that label, hybridization will be done based on these chips. Okay, so here we are mentioning Fe matrix chip. Here I have mentioned Fe matrix chip. On these chips, there will already be information on the all type of genes which are reported so far. Okay, so corresponding to that, whatever genes are available in our sample, that will map to those genes whose information are present on the chip, and that will be and the quality or the intensity of those genes will be assigned to each gene which are connecting with the information which are already present, or mapping to the information which is already present on these genes. So those information we can also use for further data analysis for gene expressions. Okay, so hope that is also clear. Next, I can show you, this is a cell file where we have the header lines. Like you can see on the top, there is cell file mentioned, versions mentioned, then there is header. Okay, so total number of columns, then rows numbers are there, and there is other informations given. So offset grid is nothing, so that's why it's given 0. And so grid and other information, this is based on the chip that is being used, or the platform that is being used to process this particular data or the sample. After that, we will be knowing here that there is the total number of cells are mentioned here that are mapped to our chip. And then we have the general information about all the genes that are mapped, or the probes, because probes corresponds to gene IDs, or standard genes that we have. So related to that, the information is given in the tabular format. These file format are again like a special type of files that we cannot just open in Excel and like perform the analysis. So we need a specific tools to read the data from this file to understand the gene expression. And for microarray data, as I was talking about the chip, you can see here we have the information with the on the chip with different colors that corresponds to the gene expression or gene intensity, we can say. And we will be having the data like data from this type of analysis, microarray analysis, in form of gene IDs in the first column, where total number of genes that we have in the body will be there based on the organisms. And then we have samples. So whatever number of samples are in your data set, all will be there in the column wise, and corresponding to each sample, what is the gene expression, that will be mentioned in the matrix format. Now we will discuss about databases. What are different databases? So I will go through it like very quickly, because this theoretically it will not be much feasible to understand this. So maybe I will be showing you practically how to use these databases. First is NCBI, which is well known database for different type of sequence data that we want to retrieve from it. Because NCBI is basically a collection of like a big database, which is collection of many other smaller databases. So we have list of all databases which are included in NCBI. Under this list, we can select, there will be the drop down list when we click on this triangle, and then it will show you all the list of databases which are included in NCBI, and we can further explore and retrieve the data. So this is information, and what type of data is there within NCBI is mentioned here, that we will be having microarray based data, so which is stored in geodatabase. So within NCBI there is geodatabase. Then we have SRA, and which will contain DNA and RNA sequences. Another database that is included in NCBI is GenBank, which will be having DNA sequences. PubMed is also there within NCBI, where different research papers which are already published are there. Okay, other databases are also there for, specific to epigenomics and other type of data that we want. So this is just a drop down list which is showing all type of databases. As I was mentioning, that geodatabase is there within NCBI, so it is here. Then we can also check different books here, because they have given wide variety of resources to understand and use for research purposes. Then we have Geo database, which is also a standalone database, and also it is included in within NCBI, where we can search for microarray based data, and or also RNA data. But RNA data will be processed data here in Geo, but we can get raw data for microarray data from this database. Okay. So we will see during practicals. I'm just skipping this fast so that we can do some practical session if we have time. And this is the interface for Ensemble database, which is again having like information, biological information related to genes. Then there are different tools included within it, such as Biomart. Then we have Blast. Then we have VEP. So VEP tool is variant effect predictor, that is used for VCF file format generation. Okay. So if you have, if you have VCF file format, if you want VCF file format from the gene list that you are having in hand, then you can use this tool. So there are different tools, then there are different organisms included, for example, humans and mouse, and there is zebrafish also, like below this. So once you open the database, you will see I have added the links of databases, so that you can directly like be diverted to the link and open the database. So once the PPT is shared to you, this is just the information. Other database is Array Express. Again, it is having like sequences and sample informations. Then it is also having the microarray and RNA-seq type of data that you can download and process. There are also process data within the Array Express. Uniprot is protein specific database, where you will be getting different type of protein information if you just give the name in the search bar, or you give the ID of the protein. PDB database is there, where again you can download the protein structures, 2D, 3D structures, or you can also analyze data here. As you can see, there are different options given in the tab. So we will see if it is required during the sessions. We have PubChem for drug specific data, and we can like if you want to explore details of any drug, maybe in the medicine you are taking or your parents are taking, maybe, so you can also explore the like details of that drug within PubChem. So this is drug specific. Okay. And then we have plant database, which is specific to plants. And since this is not our much concern, but I'm just sharing all types of databases that we have. And this is not the complete list. Then we have Uncon database, which is again for plant specific, and plant list, which is also the plant specific. So whoever is interested in plant genomics, they can proceed with this type of database. We have Branda for enzyme specific database, so that we can this is used in genomics data analysis at the downstream step, when we further want to explore the functional parameters for the genes that we have identified, or we want to identify the metabolic pathways where our genes are involved, the genes that we have identified during gene expression analysis. So we can use this database as well. Then some cancer specific databases we have Cosmic, which is catalog of somatic mutations in cancer. And then we have ICGC, again for cancer specific database. We have NCG with there are different versions of NCG, so you need to check which is the updated version so that you can work. Always work with the updated database. So whenever you are working with any database, must go to the information or about the database page and see that when last it was updated, so that always work with updated. At least it should be updated within 5 years, so that you will see that all type of informations are there even from latest researches. Okay, these are some pancreatic cancer specific database. We have cervical cancer specific database, and we have some links where you can specifically go to databases which are important for. So CG Hub is for huge data that you can use for your analysis. And we have Cosmic and all the different type of databases I have included. So if there is any query, let me know. Otherwise, let me check if we have how much time we have, so that accordingly I can share my Chrome page for some practical session. Yes, we do have time. So if there is any query, can you please let me know? Accordingly, I can proceed.
Kasala, can you see a question in chat?
Yeah, do we have questions?
Yes.
Okay,
I have
let
a
me check.
question from Sahana asking 1 pixel is equal to 1 probe.
Okay, so we have one question from Kirtana. What is the difference between CDS and exon? And okay, so CDS is basically, as I mentioned, that coding section or coding section on the DNA, which will be further translated into proteins, and that will be functioning for like normal functioning of the body. And while exons are the segments of the gene that are included in the mature RNA. So that is the messenger RNA, mRNA, where we are using mRNA gene expression. We also perform analysis for that. So exon actually include all those sequences which are transcribed into RNA. Okay, and CDS are those which will be translated further into, like surely translated further into proteins. So that is one basic difference between both. So while CDS and exons, they are related, I understand, because we know that after transcription, we get the mRNAs, right? But after translation, we may not get all the part of mRNAs that we are having, that will be surely translated into protein. So all those section of mRNAs which will be surely translated into protein, only those will be called as CDS. Okay, so coding sequences. Okay, okay. Repeat the difference between GFF and GTF. Okay, and then yes, 1 pixel is equal to 1 probe on the chip that we have, Affymetrix or any microarray chip that we are using. Let me share you the screen with I will show you screen. Okay, so Where is my screen? 1 minute. I'm sharing it with you. Yeah, so first I had to share you the PPT. Where is the, okay, this is the PPT online. Let me just go back to, because visually we understand better unless we have the files in hand. So that's why I'm going back to slides to explain you the difference between GFF and GTF. Okay, so here, as I mentioned, that in GTF file, we have the information regarding the like location of the exact location of the genes that we are having in hand or that we have identified or the sequencer has captured from our sample. Okay, so we have all the information about chromosome and the source that from which organism or maybe which platform it is obtained, and then we have the type of that particular gene that is like retrieved, or the the DNA sequence that is retrieved at the from start to end position. Okay, so starting from 234 to 1543, that long sequence is captured on chromosome 1 and it falls into exon category or exon section of that gene. Next is the scores are given. So here it is missing because maybe the tool is not having information of this particular sequence which is obtained at from 234 to 1543. Totally depending on the tool on which the data is processed. And then we have GFF file format. So both are kind of similar file format because we can see we have similar type of information and all the columns. One additional thing that we have in GFF file is strand information. So here we can see in GTF we didn't have the strand column, but in GFF we have the strand column, and sometimes strand information is important. We will see in databases right now when I will show you on web that what is the strand actually and why it is important to understand, because for when we know the strand information that whether the gene is going into positive direction, whether 5 prime to 3 prime direction or 3 prime to 5 direction, accordingly we can understand the direction of gene and how it is going to impact the other genes which are beside them. Okay, so that is important in some cases to know the strand information, and we need to explore that, and having that in our data is better, because more data we have, more insights we can retrieve from the data in hand. Okay, so hope that is clear. Now I will move to okay, sorry, now I will move to Chrome. Let me close this. Where is Chrome? Yeah. So this is Ensemble browser, but before that, I should show you NCBI. So when you will search with NCBI, you can also search with the complete name, National Consortium Center for Biotechnology Information. Okay, so here is the search bar for us. And here is the list of all databases. When you click on the uh, downward triangle that we have. So all the list of databases are here. You can see for after dragging, and here is SRA. We also have toolkit. We have PubChem where you can see the drug related information. We have protein specific databases for protein clusters, protein family. And then we have other databases, for example, if you want whole genome for any specific organisms, you can go to genome. You can retrieve microarray data sets from geodata sets or geoprofiles. Then you have dbware and dbgap databases, where dbware will be having the variant information, and variants are basically the mutations that we talk, that the reference nucleotide is different, and then altered nucleotide is different at the same chromosome and same location. So that type of variations are captured in DBWare. And then we have biosamples, books, clinical variation informations we have, okay, and bioprojects. So projects are will be having different type of samples composed of these databases consist, okay. So we can search for any data set. For example, you are interested in lung cancer, so let's proceed. Okay, so that search we can see, but you can also see here in interface, we can submit the data. For example, you are working in wet lab, you can submit the data, you can download the data from directly from here. You can, there are learning materials here. Then you can you can develop tools by using the NCBI API, and there are tools to analyze the data as well, and research specific tools, okay. So further you can like explore this database because it is a more comprehensive type of database which includes many other databases, so kind of very comprehensive. Now we can see how we can search for the data set. Here is the lung cancer I have added as a keyword, and when I hit the search, I can see here that what all information is there within NCBI, and we can see a lot of information here. But we need to know that what is our objective and what exactly we want. If we are interested in genes specific to the lung cancer, we can hit at this particular option where we can see 6,000 informations are there. Then if we want the data set from microarray, we can click here and it will redirect us to that link, okay. So you can also do open in new tab so that you can also explore further information on this page. Otherwise, you can directly hit it. And there are different options, proteins related. So here are the proteins related to lung cancer. And we can also have DNA and RNA specific samples under SRA database. Okay. So SRA database is specific for RNA and DNA sequences. Clinical details and related data are all included within DBGAP as DBSNP and other databases which are there in NCBI. Okay, so a lot of things to explore. You can go through one by one. Let me just go with a geo data set. So we have total 75,000 information. So I'm clicking here, and now we can see the interface where most, okay, so where we have the information about the lung cancer. So every sample which we are looking at here will be having the keyword lung cancer that is highlighted, okay. At the end you can see here we have lung cancer, and that is highlighted because you have searched for lung cancer. Okay, there are other filter options you can check here, that if you want platform specific data, for example, you want data from Affymetrix platform, not from Agilent or other microarray platform, then you can also filter that. Okay, so you can click on platforms and it will show you platform specific data and plot platform related. So now you can see the data set is from mouse and the platform is Affymetrix. Okay, so this is how we filter data. Also, we can filter based on organisms. So here we can customize that which organism we want. And then we want expression profiles. Let me just zoom it because that will be more clear. Okay. Okay, so now we can also filter based on study type. We want to do expression profiling or gene expression analysis, or we want to perform methylation profiling. Okay, so methylation profiling will comes under epigenomics studies. Okay, that will be based on histone and whatever methylation happened within DNA. We want tissue specific information or other type strain specific information, that whether you want to check for data set which are related to publications that are like published and reported within 30 days, like last month or 1 month away or 1 year. Okay, so this is how you can filter. Other type of filter are on right side, where you can see directly you can filter for different organisms. And right now, if we see the search results, we have total 75,578 pages for lung cancer data. Okay, so for lung cancer keyword, as we have searched here, we have 75,000 total pages, and on each page we have total 20 samples or 20 results available. Okay, so these are total 75,000 samples exactly, but we have 3,000 pages total. Okay, so that was like mistaken for it. So we have 33,000 pages. Okay, 3,077. But now we don't want for mouse species. We don't want for other type of Drosophila or other species. We just want for Homo sapiens. Okay, so out of 75,000, we can see that out of 75,000 samples, we want just for Homo sapien, and this should give you a filter of only 60,000 samples. Okay, so let's just click on here so that it will select only Homo sapien samples. Now you can see, as we have filtered, it has modified our search bar with the tag and. Okay, and you can also manually add this type of tags, like with lung cancer you can mention lung cancer and Homo sapien. Even if you don't know the ID, taxon ID, which is TX ID for the organism, you can directly mention Homo sapiens here. For example, you are not going for like filter here, then you can directly search like this, lung cancer. And so must remember, this and will be always uppercase for to filter here in NCBI. And when you search, then it should give you only 60,000 samples out of total 75,000 samples. Okay, and accordingly, if you go one by one on each sample, so you will explore that what type of data is there. What is the notice here? They are giving alert for maintenance. That is starting from July 25. Okay, that's not problem. They will hardly take. Okay, so service will be unavailable for 24 plus hours starting from 9 p.m. That's quite long. Okay, no problem. We are already having a lot of data. Even if the server is not available, by the time we are going to start the practical session, we have the data, we can start analysis. If the server is available, we can directly download the data from these databases and proceed for analysis. Now, this is the data set within Geo, because we tried to explore, and on the top you can see the gene expression omnibus, that is the Geo database full form. And we have the samples, this dataset ID, which is GSE, the series 297846. You can see the details when it was submitted. So very recent data from 2025, July 17. And the title of data is related to tumor microenvironment from small cell lung cancer to immunotherapy. So treated samples. And other details you can also check here, and organism is Homo sapiens, as we have already filtered. Details and the summary of the experiment is mentioned here. So this data is submitted by the wet lab scientists, and their experiment details is mentioned here so that we can understand the data better, from where it is coming, what experiments has been done on it. Accordingly, we can select this type of data. Overall design is also mentioned. And then there is interesting information that we always look while working on data, before even starting working on data, is whether the study is published or not. So first thing, we try to look for where when it was submitted, like what is the status of the project when it was last submitted. It should be a recent data so that it is not published like more. or more work is not published on that. And also if it is published, then you should go to the link of the paper. And if it is not published, then there will be no paper mentioned here. They will ask that if you know any paper which is associated with this particular data set, then we can mention. Okay, so we can notify and they will add it here. But since it is very recent data, the no work is already published, so you this seems a good data to start analysis, okay, on this. But we can see here in the bottom, like after all the details, you can see details of submitter, that email is given. Okay, so this is also important. Sometimes we see this as unwanted information given. Why it is given for us? Because why we want to contact the like whoever the person who has submitted, but it is required in some cases, and I will let you know when and why. There is other information which is platform information. This is very important because depending on the which platform this data is processed, you will be selecting the tools. Okay, so this is important. You also need to see how many number of samples are in your data so that accordingly you can proceed for analysis. And we have bioproject information where you will get RNA-seq data for the same data set in NCBI repository. So you can also redirect from this link for the project if it is available in the NCBI. Probably it is available, that's why link is given here. And here at the very bottom, you can see the raw data information is given here. So for this data set, raw data is available here, whose size is 14.5 MB, and it can be downloaded from here. But right now, what you need to see is file type is tar, that is the zipped file, okay, compressed file. And but within tar there is text file. So whenever we start analysis, we always prefer to have a raw file, okay, raw data only, because if there is a text file for microarray, it means it is processed somewhere. Either the data is normalized or processed by the submitters who have submitted this data. So this will not be very much useful for us or to claim that this whole study is done by us. Okay, because the data you are already getting a processed data. So one step is already done by someone else. So that is not totally your study, right? So that's why you need to know that whenever you are downloading the data, your data should have cell file format. Okay, so that is one important thing. Other thing is why you want to contact the submitter or email is given, because somehow you find this somehow, or in some cases the disease is rare and the data set is very rare in different databases. So if you want to work only on this data, so in anyhow or any case you want the raw data, right? So but in this database the submitter has not submitted, so you can contact to the submitter through this email ID that I need a raw data for this particular experiment that you have performed. And you can mention your requirement, why it is really important for you, and if they want if they are convinced, they may share you the raw cell files. Okay, and that can be helpful for you to carry out the results. But you should not randomly, like anytime you are not looking at, like cell file is not present, you are emailing them again and again. Don't do that. If there is alternate data already available, then you should proceed with that data. That is very rare case when we have to email these submitters. Okay, so that is just an option. Next is ensemble data. Ensemble database is another database. I have just shown you the interface for ensemble database. Okay, so this is the interface where we have the list of tools mentioned here on the top, and then we can download information from here, like list of genes and other informations. We have documentations and related blogs and the tools here. Directly we can apart from that, we can also search for different species that are included within this particular database so that we can download their related genes if you want to see. For example, you can see they have mentioned BRCA2 gene if you want to check in Homo sapien maybe, so humans, and you can search for it. So they will provide the related information. on this, but I'm not doing this because now I have to explain the things here. And we have different genome informations here, which is humans and mouse and zebrafish. Then we have additional information for pig breeds also here, and other species. We can see the list. And reference genome, this is one of the database which we most prefer whenever we have to download the reference genome to map our samples. And we have the latest reference genome which is in use by researchers is GRCS38 for Homo sapiens. And if you want to use the previous reference genome, then that is GRCS37. So this link will redirect you to that reference genome, and you can directly download. There are other options to compare the genes across to find the SNPs, mutations, and alterations in your genome sequences, gene expressions in different tissues you can see, and the gene sequences you can retrieve. Okay, so that is your task to explore these things so that and make the note so that whenever you have certain tasks, you should know that which database you have to go. Okay, now I will show you for one gene, like I'm searching for BRCA1. That is one gene more very widely identified in different type of cancer, including breast cancer, ovarian cancer, and other type of cancer. So we have the gene information here. We have the transcripts, variants, and other type of information for this particular gene. We will try to open the link so that I can show you the strand information. That was the query related to difference between GTF and GFF. This will all clear out here. So this is the gene that we have, and each database have their specific type of IDs to for searching the gene. Okay, so BRCA1 we have used the keyword and the database has already returned the answer. But sometimes what happens is the database will not recognize BRCA1. It will recognize only the ID that it uses. Okay, so for genes, ensemble database will use ENS for ensemble, and G will always be there when you are searching for gene. When you are searching for protein, you need to mention ENS and P. Okay. And when you are searching for transcripts, it will be ENS with T. Okay. So that is the difference for ensemble gene IDs and other type of IDs that ensemble database uses. And then we have the information, the description of the data of the gene that we are searching here. And then we can see here the HGNC database is having this for the same gene. If we want to search in HGNC database, then we need to use the keyword 110, not BRCA1. Okay, maybe it can give result with BRCA1, but sometimes databases don't give the results. They are specific to their IDs. Okay, so accordingly we should be aware of these IDs, but if we don't, then we can always come to different databases, for example ensemble, which is widely giving different type of IDs and also along with the general name of the gene. Okay, so that will be helping, and there are other tools that can give other gene synonyms is given. The same gene, the BRCA1, can also be sometimes in publications it is mentioned as BRCC1. So you should know that BRCA1 is also known as BRCC1. Okay, so if they report that BRCC1 is involved in this particular disease, you should know that this is the same gene which we call BRCA1. And then there is FNCS and other names for the same gene. Okay. So during literature survey, this is very helpful, the gene synonyms, because sometimes in papers they don't mention BRCA1, they will mention RNF53, and when you will be reading the paper, you will be thinking that this is some other gene. You are totally not aware of BRCA1 involved in that particular disease, right? So we should know at least few of the synonyms so that we should not misinterpret while doing literature survey or any type of analysis. Now here the location is mentioned where the chromosome number is given. So on chromosome number 17, the BRCA1 lies, and particular start and end point is given as 43004 295 up to 43170 247. Okay. Now we can see here that here it is mentioned as reverse strand. It means it is on the negative strand. That's why it is mentioned as it is on reverse strand. Okay, so this is important because based on that only the gene location, the start point and end point will be deciding based on the whether the gene is on reverse strand or the positive or forward strand. Okay, other is this gene consists of total, so this gene is consist of total 41 transcripts. Okay, so transcripts is combination of the reads that we have in the gene or the sequencer has captured, and here we can see the transcripts information. Okay, so this is the table for transcripts. When you open the table, and there is certain information, if you don't want some information, you can also hide. So if we unclick, then our table get shorter. For example, we don't want uniprot information right now because we are not dealing with proteins. As of now, we are concerned about gene. Then we want to know that whether the gene is coding sequence region or not, then we need to keep it. Then about the biotype, whether it is protein coding gene or not, then we need to have it. What is the translation ID? So, and translation ID we will not be interested right now because translation is about the proteins, so we will remove it. And protein ID also we don't need, or we can see that what proteins are generated, but that is not our concern right now. We can see what how long the base pair is or the sequence is for this particular transcript and name of the transcript. Once the table is sorted, you can also not unselect, just keep it as default, whatever is mentioned, but just to sort the information and remove unwanted information that we are not focusing here, we can see different total number of 41 transcripts as they have mentioned and their combinations. Okay, so this is kind of information. So now here we can see at this particular stage, that's what I was about to show here, that some of the transcripts will be from protein coding sequences and some will be not from protein coding sequences, the other sequences. So nonsense mediated decay, nonsense type of mutation, there are different type of mutations. So here different categories are defined. Then another transcript where we can see it is protein coding CDS. Here protein coding CDS is not defined, coding sequence is not defined. Okay, so different type of categories are already mentioned here, and we need to focus, like if you are working with coding sequences and only those part where we are focusing on coding sequences, then we will focus on only those transcript which corresponds to protein coding sequences. Okay, so this is one information that we have in ensemble, and other details we can also get for our gene that we are searching based on whatever information we want. And there are annotation attributes also given that where they are involved. Okay, so here we have visually we can see different information for the gene, their transcript IDs. Their transcript IDs are mentioned, gene IDs are mentioned, what are their regulatory features, at which particular locations the regulatory features are there, which may be regulated at particular like gene expressions. Then we have protein coding sequences, all are highlighted in the figure. If you want to explore more about your gene or understand how where exactly it varies, then we can see, and different information is all given here. You can also directly go to the different information from the tab which is given here. So this is about the ensemble database. Let us just quickly go through Array Express because that is also one of the database we use most of the time for getting the samples. So we need to search here, different options again given here for the services they provide, they provide training, different type of trainings they provide, even virtual trainings or also on-site trainings that they have in UK and other centers. So you can explore that. They provide really good trainings. And then now we have, okay, so we are interested in lung cancer. Let's just assume, don't get scared, because some of us like get really scared when we hear cancer, right? So let's just explore about lung cancer. Yeah, so now we have the list of data sets here and the samples. We can see total we have 1,000+ results, which belongs to lung cancer. And again, we have filters on the left side where we can filter if we are interested in chip-seq data or DNA sequencing data or RNA sequencing data, then we can filter it out. Okay, so whatever data we are interested, we can filter so that we can keep the list short, because we have to ultimately select any data set to proceed for analysis, and we have certain conditions to select the data set based on number of samples and the type of files available for analysis. Okay, so if we want to select that all, give me all the data set which have raw data available. Okay, so we will select this option. Now it will filter only those data set which have raw data available, not the processed one. Okay, so now we have like samples only present about 1024. Earlier it was 1065, right? Or 64 maybe. So here we have the raw data samples only. We can click on any link and proceed to see that what samples are present there. Okay, so apart from the details that when it was submitted on 2024 November 19, and like when it was again modified on the next day, very next day, 20 November, and how many people have viewed this data, okay, or the data set. So these all informations we can see what is the detail, who are the authors who have submitted, and what is the data about RNA-seq for coding RNA from single cells. Okay, so T lymphocytes. So the description is all given here. We can go through if we are interested in this data set. We can also see the publication. Since it was submitted in 2024, we have the publication, and we can go to the link of this publication, or maybe we can search based on the authors and the title of the paper. So title of the paper is here, as they have added the citation. So all authors list are given there, and the paper topic is given here. The title is given here. You can search with this title based on the first author name, so that you can get the detail that what the paper has discussed related to this data, and how differently, if you are going to analyze the same data, how differently you can present your results. Okay, maybe you have some different idea to present your result for different idea for doing the analysis, so that you can do. And detail of the samples we can see total number of sample counts. We have 27. What is the experimental design, organism, and everything is given there. So I'm not explaining all of this here. You can further explore data files we have here. So maximum we can try to see, because we have 27. So it should provide us 27. So here, this particular AliExpress will give you samples in the with the keyword EM tab. Who is annotating this? Please don't do. So we have EM tab format data with IDF and TXT format. And now we can see here, and we can download these files. Also, we have the link for ENA database. The same data set is available in ENA database. ENA database is basically used to download the raw data. Okay, so if we have the link for ENA database, then it is recommended to directly go to ENA database and download the samples from there. Okay, so that we can do. And apart from that, what we need to, we can download these files which are available right now, because we can see they have mentioned there are total 27 samples. So sample count is 27, right? So we can download all the files from here, download all files, and download as zip. Yeah, we need the zip file. So it is downloaded, right? So now you know how to download the files. And yeah, so I will show you quickly with ENA how. So this link I have just directly hit on the link, and let me go to ENA. This is ENA database, European Nucleotide Archive. And here, this is the same ID, which was the dataset ID, which was just we were exploring. Here is the dataset ID on the top, or even here also ERP 156718. Otherwise, ID is already given on the top, maybe somewhere. So this is based on experiment, but but the process ID we have already here. So now we can see below that what samples are given, and we have the samples given here. The information or the samples are all given here, but we don't have the FASTQ files. So we need to go check if we have FTP files. They are not provided actually, maybe due to some reasons. Okay, so if they have not provided, what we can do is we can directly download it from here, as they have mentioned that we can download the files. Maybe they have compiled them all in the two files only that they have mentioned here, and as they are text files, so we can directly open and explore them. Okay, so we will deal with the data sets and how to open and explore the data, that we will do later. But right now, today we had the session for exploring the databases. So that is the databases. Now we can see some of the other databases, so that you will be aware if anytime you have to go for proteins, because anyway we are going to explore different genes. So if we want to know what protein is going to form with the gene and what will be its properties, and for example, if gene is mutated or any nucleotide is altered on the gene, then what will be the impact on the corresponding protein that is going to be translated from that particular gene. So that we can search here. Maybe we are, if we are interested in drug related protein, so we can search for that. And here we can see we have different genes which are related, different proteins which are related to BRCA1. And we can like open them if we are interested, like we can open them and see that what are their 3D structures, what is their details, and whether they will be like helpful in any way to further provide insightful results on our data. So, and there is a paper already published for this particular protein, so we can also correspond to that protein or to that paper, so that we can better understand the particular structure which is provided. Other information that we have for protein is about the total is weight, and then atom count, and other information that we generally use or more concerned when we are dealing with the proteins. So different informations for your protein is mentioned here. Anytime you are thinking about protein or you want to explore, you can come to this particular database, RCSB PDB, and you can download. Even you can download, for example, sometime we go for docking and simulation, so we need the 3D files of this particular structure. So you can download that in PDB format or maybe CIF format if PDB format is not available, or and convert the file format whatever is required. Okay, so structure summary is there, then we have the structure information. So directly it will also redirect us to the different section of this particular gene. it is loading. So figure will also be loading. If you want to see the structure only, so you can see the structure of your protein, you can move, you can see like how your protein looks. And when you will see the sequence actually, so this is not the nucleotide sequence. These are amino acid sequence on your protein. Okay. So corresponding to your gene, you can see that what particular location that particular like alteration or mutation has happened, and corresponding to that you can map it to the amino acid sequence and see that in which particular location on your protein that mutation has happened, for example this particular part, or where exactly. Okay, so you can also zoom the figure and see that where exactly the changes are happening in your protein and how they will be impacting your protein. Okay, so these all will be covered in the like are covered usually in the molecular docking or CAD related projects or courses. So, and extensive genomics studies that we do. So that goes into proteogenomics studies where we are also concerned about proteins apart from genes. Okay. Now, one another database that I should explain you quickly is okay, is PubChem that I was mentioning for drugs. Okay, so this is a specific drug-based database, and here you can search for any specific drug or maybe any gene, so that you can see that whatever any type of alteration happen in this particular gene, what type of drugs are prescribed, okay, to the patient. So you can also search gene specific, but if you are interested in any drug, so let's just take example of, no, just a simple, just a minute, sorry, aspirin. So aspirin is one medicine or drug, we can say. So don't like confuse about the this is different type of drug. This is just the medicines, okay? Formulated medicines. Okay, so this is the drug. Here is the information given. What is the like PubChem ID for this particular drug? If you want to search, what is the this molecular formula for this drug? What is the weight of the drug? Other information that we use in CAD related studies are given. The SMILES structure for the drug, and also there is other information that you can see for the drug, that is what substances are included, like what are other drugs which are involved or included with aspirin, or where aspirin is included, what are the different pathways where the aspirin is associated, so that the pathways which are maybe changed due to some disease, so we provide them or we prescribe them aspirin. The physicians prescribe them aspirin to like to rectify the disease and something. Okay, and there is literatures, bioassays are related to experiments that has been done to prove that aspirin is like effective in particular disease or treatment of disease. Then we have the literatures or the publications that are associated to prove that okay, these are the particular information that we have mentioned in this database, which are taken from the particular publication, and what are the patents filed for this particular uh, medicine or drug, we can see. Okay, so all the this type of details are given. There are many other databases also related to drug even, or proteins even, so that we can check along whenever they are required to use. But right now this is, I think these are sufficient to get started with the analysis or the for the hands-on practice, because from here only, from database only, we are going to retrieve the data sets and then proceed for further analysis. So that is sufficient, I think, as of now. As we go along, if any database or any tool is required, I will let you know so that we can explore together. So during class, you can understand the same thing which I am explaining. You can do at your machine, and I will be doing here. Okay, and you can view. So if there is any question, let me know if it is clear, like everything, because this is very easy just to explore the interface. It is your task to go and go through all these databases so that you will be knowing that what information is present and on which page and in which database, so that in future whenever you have to use any database or retrieve any information, you should be knowing that for drug information you should not go to ensemble, okay, or ncbi. You should come to pubchem, or even zinc database is there, or drug bank is there. So you should go for a specific task, you should go to specific databases only, not to the other database which are having the sequencing information only. Okay, so that is why we need to be aware of these type of databases which have the information related to the specific details that they have and which we want for our objective. Okay, so hope that is clear. Let me know if there is any query, and if not, then probably by tomorrow we can start the hands-on practice as per the schedule. Okay, so is it clear? Can you please give me a real quick yes so that I can understand? Um, oh,
Dr. Dasana, we have few questions in the chat.
yes.
Yeah.
Yes, okay. So Sahana is there. If I have list of VCF files from variant calling pipeline, using this files, what kind of analysis that I can do for better understanding? Okay. So if you have VCF files, you can understand that whether the mutations or alteration that you have identified, because we have just seen in VCF file what type of information is there. We have like reference sequence or the nucleotide and the altered sequence or the nucleotide, right? So we know that there is some certain alteration happening. Now we need to go for the tools that can determine that whether this alteration is reported anywhere in any paper or any database. So those tools will be having these information. Okay, so they will just map your data, your alteration list to those data, to those information, and will tell you that this alteration is related to certain disease, if they have been reported in lung cancer or not, or any other disease maybe, or whatever number of diseases they will mention. They will also give the score of being that mutation as deleterious or not. Okay, so if the mutation or the alteration is deleterious type, because these tools, there is oncotator and other type of tools which can use these VCF files and can mention you more details about this information. So they mention some terms, and you need to understand those terms. They details are all there within the tools. So for example, if they are mentioning that this particular mutation or alteration, whatever happening, is deleterious type, it means it is a disease causing. Okay, if they mention that this is benign or not defined, not sure, okay, so you can say that this may be causing the disease or not. But if they are mentioning that it is deleterious, you are sure that it is causing the disease or any type of abnormality at the DNA level that is causing or that may be playing the role in the progression if this of this particular disease. Okay, so VCF files are important, and we can further explore the more insightful results and comprehensive results from them. Next is from Vishwas. During NGS analysis in step of alignment, for example, alignment by BWA, after that we get SAM format file. Most of people prefer convert SAM format to BAM file. Why? If we continues with SAM format, for downstream analysis, it might be get different results. Okay. So first part of your question is why we prefer to convert SAM file to BAM file. Because SAM file is again the tabular data or text data, right? And text data with huge amount of data, a lot of sequences, a lot of reads are there. We cannot interpret the result or any kind of finding from that, right? So we convert that in binary format so that the available tools which are already there, we can use them we can use the BAM file in those tools to visualize the data, to see the same text data which we were not able to conclude. We can see the difference, like just how I just shown you in the PPT, that on above there was disease samples and in the below slides we had the healthy samples. So we can see that there is certain nucleotides present in healthy in the diseased patients, but not in the healthy people samples, right? So we can see the clear comparison between the two samples. And based on that, if for example, if we are interested in just one gene, okay, let's say BRCA1. So we will focus on BRCA1 slide or section only, and we will try to see the difference between the two samples, how they are varying. And depending on that, we can note down those nucleotide sequence, and then further from VCF files or maybe from differential gene expression analysis, we can further see that how the gene expression is changing based on what we have seen in the files, right? So just for visualization, because as we know that visualization supports a lot of results, and can, like, one picture is worth more than 1,000 words, right? So that's why visualization is very important, and that is the only purpose to convert the SAM file into BAM file. If we continue this SAM file, so results will not be different because information is exactly same. Nothing, not even a full stop is different in from SAM to BAM file. Okay, so result will be totally same. Why BW aligner generates SAM file? Why not BAM file? So SAM file is process of analysis. Okay, so it is supposed to be in text file because anyway we are providing FASTQ file, and FASTQ file is in text file, right? Spam file we are just generating for our use, okay, for our understanding, okay. And according to that, because most of the scientists and researchers prefer to have BAM file because they want to visually see what is happening, how the samples and their expressions, gene expressions are changing, why they are changing, how much they are changing, and at what particular location. So they want to see. That's why the further tools which are there for downstream analysis and gene expression, DEG analysis, differentially gene expressed analysis, they prefer to see the BAM file and information in BAM file. That's why they have made or developed the tools in such a way that those tools will take the BAM file as input and then further proceed for DEG analysis. Okay, so that is one significance. Swanti question is, for PhD and master's dissertation, is it better to start from a raw pre-processing, or we can instead integrate count matrix from different experiments and proceed? Okay, so this is very common question which everyone asks, that can we start with the process data? We can start, okay. But in case when samples are nowhere present, like if you are interested in lung cancer, so if lung cancer samples are nowhere present in any database, and you are interested in that type of data only, lung cancer processing, data processing only, so in that case only if process data is available you can proceed. But for that you have one extra step to do. You have to go through all the details or the description of that data set and understand that the researchers who have submitted the process data, what procedure they have used for normalization, or even what type of processing they have done. That's why after that they have generated this process data and submitted to the database. Okay, so but always prefer, if the raw data is available, always prefer to start with the raw data, because in that case you will be knowing each and every step. You will be fixing or altering the pipeline of your analysis as per your requirement, and your analysis will be complete end to end by your hands only. Okay, so by your preferences only. So always prefer raw data. If no way raw data is available, then use the process data. What is the difference between forward strand and reverse strand? Okay, so this I have already mentioned that I think this is okay. Yeah. So the forward strand genes will be having the nucleotide sequences from 5' end to 3' end. Okay. So going into forward direction on the chromosome. From left to right. Okay, but the reverse strand genes on the reverse strand will be going from left to, their sequences will be read from left to right direction, and they will be calling as genes on the reverse strand. And uh, so their sequences will be in order from 3'. We will start to read their sequences from 3' to 5' end. Okay, so hope that is clear. And if it's still it is not clear, then I will add I think it is clear, Satanshu. Okay, that's good. Do we have any query? Okay, no, I think that's quite clear for today's session. And still, if anyone has any query while exploring the database or anything from previous sessions, so you can mention that in our Telegram channel. I need to check because I haven't checked that yet. So yeah, I will answer that there. Okay. So don't think that in class or session only you can ask any question. You have any type of question, you can drop there in the Telegram channel. I will answer that. Okay. Okay, so
Thank you,
I,
Dr. Dasar.
yeah, yeah, thank you so much. I think we can wrap
Yep.
up the session, right?
Yes, yes. I was about to say the same. Dear participants, if you have any questions, kindly post it in the Telegram discussion forum. The recording of today will be uploaded in the classroom shortly. Let's meet tomorrow at the same time. Thank you all for joining today. Thanks a lot, Dr. Asad.
Thank you.